Hi everyone, welcome back. This week is our second last week of new material where we're going to be focusing on chapter 7, looking at evolutionary theory. Now, the study of ecology describes how organisms live in their environments. Arctic foxes have adapted to live in environments where there's snow, as well as times of the year where there may not be snow. Remember, we talked about the heat-sensitive alleles that are able to turn on and off the production of fur pigment. And so here you can see that the heat-sensitive allele is turned off, which then will shut off the production of pigment, and as a result, the Arctic fox's fur color stays white. And that helps them to then camouflage within their environment and will allow them to be more successful to escape predation. And here we have a fox that's adapted to living in more of an arid type of environment. And you can see here that's represented in the fur color as well. So ultimately, evolution is the consequence of the environment in which these organisms live over time, okay? So from generation to generation, passing on alleles that allowed them to be more successful in surviving. So two terms that we need to make sure that we can identify the difference of at this point are adaptation versus evolution. Right now, adaptation occurs at the individual level, whereas evolution is going to be occurring over time within a population of organisms. So with adaptation, we're looking at here the regulation or expression of specific genes that are going to allow that individual to be better suited for the type of environment in which they live. So we can see adaptations even occurring within ourselves. Individuals who live at higher altitudes where oxygen concentrations are lower, they tend to have an increased hematocrit. And hematocrit is a measure of how many red blood cells you have in your blood. So the more red blood cells that you have, the more hemoglobin, and therefore the more oxygen that you're able to carry. So in individuals who live at higher altitudes, we see that they have increases in red blood cell production to be able to take as much of the oxygen out of the air and take it to the tissue cells. So with evolution, we're talking about the passing of alleles. Remember, what are alleles? Alleles are the traits, right? The variations of the traits. So if we look here at fur color, the heat-sensitive allele here that would be passed on to this Arctic fox's offspring would then allow them to be able to adapt to that environment as well. So it could be that at some point in evolutionary history, there was a mutation that occurred, but that mutation was actually that heat-sensitive allele. And so as a result, those that had white fur in the winter were going to be less likely to be picked off by predators, would be able to live and successfully reproduce and pass on that heat-resistant allele to their future generations. Now, all organisms have the capacity to over-reproduce, and we can see that here looking at sea turtles. And the idea there is that within the animal kingdom, they're wanting to produce as many offspring to give the best chance of having the strongest set of specific traits and characteristics that will be able to survive and then pass on those alleles to future generations. Now, this is an extreme example of over-reproduction, but we can also see that within human populations. Maybe not so much now in human history, but even just going back a couple of generations. I think of my grandparents' generation. Even just 2 generations ago, family sizes were much larger because we didn't have the same vaccines available to be able to prevent against lots of different diseases that would tend to decrease an individual's life expectancy. So we see differences, right, in family sizes. We have those choices to make, but ultimately, all have the capacity to over-reproduce. And so what happens is, through sexual reproduction, remember we're having those individual haploid gametes that are formed. And what is the whole process of meiosis doing? Producing genetic variability. And so over many generations, there are so many different combinations of traits. When you combine the very genetically variable sperm with the genetically variable eggs from a variety of unique individuals within a population. And so there's always new combinations of traits that are going to be continuing to emerge within all populations. So if you look at this over a long-term time scale, as environments change, it will be the individuals that are best suited to those environments that are going to be capable of surviving and passing on those traits successfully. And this is why genetic variability is so important, because if everybody was the same and we had some sort of huge environmental change and none of us were adapted to that, our whole population would be wiped out and the species would be unable to survive. And Darwin's theory of evolution coined this as the survival of the fittest. Now, I included this image here because you might think, when you think fitness, or even, okay, when you think fitness generally, you're thinking of strength, stamina, endurance, and all of those things are in and of themselves great. But the idea here of the survival of the fittest, to be the fittest individual in a population, you are born with traits that are most beneficial given that specific environment into which you were born. So it may not always be that the bigger, the stronger are necessarily the ones that are going to be fittest for the environment in which they live. So ultimately, the fittest individuals, those that are going to be best suited for the environment in which they're living, they're going to be able to live the longest, they're going to be able to reproduce, and thereby be able to pass on their alleles. And so what you see over generations and over time is that the individuals that are best suited to that environment are the ones that are going to survive. So let's take this example here of fur colors. So here we have a population of mice. Okay, we start off with one type of allele. Suddenly, there is some sort of mutation that occurs. And this is typically with all these recessive traits, they tend to be mutations that have occurred in a population. So this mutation has occurred where this specific allele is now coding for no fur pigment to be produced. And as a result, this individual is going to have lighter fur color. Well, look at what's happened here. In this type of environment, these mice with the darker fur color, they were camouflaged really well into the sort of muddy soil in which they existed. And so it was harder for predators to be able to pick them off. But with this chance mutation that's occurred, causing the pigment to not be produced in a lighter furred individual, well, now that lighter furred individual has actually been easier to pick off by the predators. So as a result here, that chance mutation that occurred, it actually didn't provide any better opportunity for this individual to survive. In fact, they were actually weeded out of the population. That mutation was actually a weaker mutation, which then would not be passed on to future generations. Now, let's say we have that same population of mice, but all of a sudden there's been an environmental change. All of a sudden, the area has become very arid, very dry. Well, that's going to change the topography. And all of a sudden, all of the darker furred individuals, they are not going to be as best suited for the environment in which they live. So now when this chance mutation occurs and we have a lighter furred individual, well, the predators are going to be picking off all of the darker furred individuals that existed originally. And so who's going to be the fittest in this situation? It would actually be the lighter furred individuals. And so they would survive. They would be able to reproduce, and they would then have lighter furred individual offspring. And those offspring would then be able to reproduce and pass on. And we would see a gradual shift from the darker furred individuals to the lighter furred individuals due to some change in the environment, where the lighter furred individuals were better suited to that type of environment. And so, as we just saw, predator-prey relationships act as specific selection pressures. So selection, what are we talking about? Natural selection. Okay, so the way in which the predator-prey life cycles even are going to act as pressures as to how many of that population of prey are going to be able to survive. And this works equally for both prey selection as well as predator. As the population of the lynx flourishes, now there's going to be a lot of predators. Well, a lot of predators means they're going to be eating a lot of prey. So the prey populations are going to decrease in size. But now that there's a reduction in the prey population size, there's not as much food to eat. So the predator population size will eventually start to decrease. And as that population of predators starts to decrease, now the prey populations are able to increase as they can reproduce. There's not as much selection pressure on them by the predators. So then as this population increases, now there's more food, and so predators population can increase as well. So there's always this ebb and flow, where you can see here that there is this relationship between population size of both predator and its impact on the size of the prey populations, and vice versa. So let's look here at our example. So, okay, we have a given lower predator population. Okay, because the predator population is relatively low, okay, the prey population is able to reproduce and grow in number because we don't have as many predators. But as they grow in numbers, now there's more food to eat. And so the predator population starts to increase in number as well because there's more food to eat means they can sustain a larger population. But eventually we get to a point where the amount of predators in the population are going to start to outcompete the rate at which the prey can reproduce. And now you see a decline in the prey population, but now this is going to have a negative selection pressure on the predators. Because what happens now? Well, at a certain point, you start to see a decline in the predator population. Well, now we have less predators. Well, now the prey can over reproduce again and they can start to grow their population size. Now there's more food to eat. So the predators then, there's a bit of a lag, right? But the predators can start to increase their population size. And that's going to have more selection pressure on the prey. All right. So there's this relationship that exists between predator and prey populations, where the size of one population is going to have an impact on the other. Now, Darwin's theory of evolution can be summarized in the following 6 points, some of which we've already talked about. First off was that genetic variation exists in individuals in a population. So what's that saying? It means that we all carry different alleles for the same genes, right? So we can have a gene for hair color, but ultimately one of us might have brown hair, so we have the allele for brown hair, and the other will have an allele for, say, blonde hair, red hair, okay, or any variation. Secondly, any changes that occur in the gametes are those that are capable of passing on to the offspring. So remember, if we have mutations that occur in our somatic cells, the cells that make up all tissues in the body besides our gametes, then those genetic mutations are not passed on to our offspring. So it's only the mutations, the DNA changes occurring within our germ cells or gametes that are going to ultimately be passed on to future generations. Okay, so evolution occurs through the genetic mutations that occur within the gametes. Now, gametes here for humans, sperm and egg cells, spores, pollen, okay, looking at the plant kingdom as well as the fungal kingdom. Okay, all populations produce more offspring than will survive. Okay, well, this is that case of over-reproduction, right? And the reason why, better chance of your alleles being passed on to a future generation. The offspring that survive, they have the DNA, they have those alleles, the ones that are going to allow them to survive in the present environment. Those offspring that have those alleles to allow them to thrive, they're the fittest. And so they are going to pass on their alleles to their offspring. The ones that don't will not survive. Those that don't have the alleles that are best suited for that environment will not be the fittest and will be less likely to pass on their alleles to the future generation. And over long periods of time, a population will have a gradual change that occurs within it. And this is a process known as microevolution. So microevolution versus macroevolution. Microevolution is looking at small changes occurring within a population, so specifically a species, over time. Whereas macroevolution, these are large-scale changes that have occurred over evolutionary history to give rise to new species. So microevolution, think within the type, within the species. Macroevolution, we're thinking of the creation of all different species. Okay, so here the idea is going back to more primitive ancestor and then macroevolutionary changes that have allowed for the evolution of different species. And so essentially Darwin's theory of evolution described natural selection and the survival of the fittest, and ultimately proposing theories for the origin of species. Now, another man named Jean-Baptiste Lamarck. Lamarck was a predecessor to Charles Darwin. So a lot of Charles Darwin's work was based on some of the original theories proposed by Lamarck. Now, prior to Darwin, there was no consensus among biologists. Darwin kind of brought all biologists together to create that one framework for how they would propose mechanisms causing evolution. Now, remember, again, Lamarck was looking just at physical characteristics. And so, again, did not have the knowledge base of the idea of alleles, genetic information, and how it was passed on, the idea that mutations occurring would then be represented as those physical characteristics. So Lamarck was just looking at physical characteristics of different organisms. And his proposition was that evolution occurred by the inheritance of acquired characteristics. So the assumption that based on the environment in which an individual lived, they would then acquire certain characteristics that would then suit them best to live in that environment, and that would then be passed on to future offspring. And this is the classic example of Lamarck's theory of evolution of acquired characteristics. So these are known as Lamarck's giraffes. So his proposal was that the original ancestor of the current giraffe population had shorter necks. But because of the environment in which they lived, their food was high off the ground. And so these giraffes would have to really stretch their necks to be able to go and to feed. So over time, his theory is that the offspring would acquire or inherit slightly longer necks. And as a result, then they would be better suited for being able to feed within their environment and be able to survive, reproduce, and pass on. And as a result, then members of subsequent generations would continue to pass along that acquired characteristic, that they would have to be stretching their necks to reach the leaves on the trees, and therefore would pass on the acquired characteristics of a longer neck to then give us rise of the current species of giraffes that we see nowadays. You're probably looking at this example thinking, this is madness. But remember, again, with all of these original theorists, they were just looking at physical characteristics, and they didn't have that knowledge base of the traits and how they were actually carried on, and that mutations could occur within the traits within a population. And so comparing this theory of acquired characteristics to Darwin, you can see that he was onto something. But what definitely makes more sense is that there would be mutations to the genes that would then cause certain individuals to have longer necks. Those individuals would be more fit for that environment. They would be able to reproduce and pass on their alleles to future generations. Okay, and that's where you would see that, and that's where you would see ultimately that throughout the generations, you would end up with individuals with longer necks, not because they were just acquiring this stretched neck from their parents, but because it was those alleles that code for the longer necks that would be more fit and suited for their environment. And so that's a key difference between Lamarck and Darwin. Okay, so with microevolution, according to Lamarck, it was adapted characteristics that were passed on to future generations, whereas according to Darwin, the variation of that specific trait existed. So the longer-necked individuals were born because of those chance mutations. And then natural selection, which essentially is survival of the fittest, would then act on that trait that existed because of that mutation. So we know that we have our DNA, and our DNA, certain sequences are known as genes, right? And those specific sequences of DNA code for specific proteins to be produced. And we see those proteins as specific traits. And so this new study of epigenetics is looking into how our DNA is able to be both up and down regulated. So turning on and turning off certain genes based upon their environment. You know, one example would be looking at how your diet and the foods that you're taking in and the chemicals that are coming into your body, how they are either upregulating, so turning on certain genes, or downregulating, turning off certain genes. Remember, with microevolution, we're looking at small changes occurring within a given population, right? Whereas macroevolution is looking at the creation of a brand new species. Now, antibiotics are given for bacterial infections. Antibiotic essentially defined means anti, against, and then bio, life. Antibiotic is given to kill off a larger population of bacteria, right? In the case of a bacterial infection. But remember, bacteria are capable of reproducing so rapidly through binary fission, right? It was about every 20 to 30 minutes in ideal conditions. So from generation to generation, they can evolve really quickly. Now, within a population of bacteria, they're all going to have genes that are going to provide them with more and less resistance towards antibiotics. And so let's say we have an original population here of bacteria. We have a bacterial infection. Let's say you have strep throat, and you want to knock out that strep throat with an antibiotic. So you take the antibiotic. Those with a very high resistance to the antibiotic are going to be more likely to survive. And so this is one of the reasons why when you're on an antibiotic, you need to ensure that even when you are past the point of which you are symptomatic, that you continue to take the antibiotics through its entire length of the prescription. Because what they're accounting for is that there may be individuals from that original population that have a higher resistance to the antibiotic. So by taking the antibiotic for its full length of prescription, you're ensuring that you've reduced the population as much as possible. Remember, our own immune system is capable of also taking care of bacteria and killing them. So even if you had some of these high resistant bacteria left, we can still deal with them. But remember, even if we have just a few bacteria left, these bacteria are going to go through binary fission. And so their offspring are going to be a population of very highly resistant bacteria. And so with antibiotics being prescribed so readily within a general population of ours, you end up with this production of almost a super bacteria, right? You have a superbug. And so this is one of the concerns as antibiotic prescriptions have been on the rise over generations. Are we going to start to see more and more cases of antibiotic resistance than we already do? Okay, this idea of antibiotic resistance where you have some offspring that survived, right? They're the fittest. It's not good for us, but for their populations, yes, they're the fittest. Okay, they have the highest resistance to the antibiotic. They're going to be more likely to pass on those alleles that allow them to have that resistance to the antibiotic and to be able to survive. And so this is an example of microevolution occurring just within a bacterial population. Okay, so in this case, just to add to our definition, selection pressure, remember we looked at predator-prey selection pressures? Well, here we have the antibiotic is acting as the selection pressure on the bacteria. And so the antibiotic is going to cause only certain bacteria to be able to survive to create offspring for their future generation. Now, another example here is artificial selection. And we talked about this before. I forget the context in which we were discussing this. The domestication of dogs or any domesticated animals has evolved over time through a process known as selective breeding. And this is a form of artificial selection because it's not nature that's selecting, right? In natural selection, you're looking at the environment in which they live is going to dictate who is best suited. In this case here, because it's breeders selecting specific offspring with specific traits that they want to breed, through that selective breeding process, they are artificially selecting who will be passing on the traits to the future generations. Another example of artificial selection is looking at many of the foods, specifically vegetables, as we know them in the grocery store, have been artificially selected for their specific traits to make sure that the farmers ultimately are able to yield the most crops and be most profitable. Okay, so in both cases here, both with breeding of animals as well as with farming and food production, we're looking at cases of artificial selection, okay, where an individual has been selecting traits that are most beneficial based on what they deem to be most beneficial. Now, Darwin spent a lot of his time on the Galapagos Islands just off of the coast of South America. And the Galapagos Islands are a very unique set of islands with very extreme differences in environmental conditions from one part of an island to another. And his observations were of a variety of finch species that were all very similar to one another, but had some differences, primarily between beak size and shape. And that beak size and shape had to do with the type of food that they would be sourcing. So birds with long bills, they would be able to open up cactus fruits, feed on the fleshy pulp deep within. Those with much shorter but larger beaks would be able to strip bark from trees and be able to eat insects with inside. So much larger and stronger, but also much shorter. And then those with intermediate bills, they would be favored during non-drought years, where many food types were available because there was so much rain and the environment would have been much more lush. And so Darwin's theory here was that microevolution within different populations across the islands would have eventually led to macroevolution. Because remember, if you have a population that's going to be changing and evolving to its specific environment in which it lives, and that population's isolated from another, they are going to be independently evolving in an event. And ultimately, if the environmental conditions are different enough, you can see the rise of a new species. And then ultimately, those generations would be passing on specific traits that would be uniquely suited for the environment in which they live. And so you would see macroevolution, so the evolution of a new species occurring over time. Now, there are two genes that play a major role in the development of the shape and size of the bird's beak. One of those genes that makes the larger beak codes for a protein known as bone morphogenetic protein 4. The other produces a protein known as calmodulin. Now, changes in the expression of these specific genes that will be producing these proteins would then be causing for certain individuals to be produced that would have certain beak shapes and sizes given the environmental conditions. And so he found, even from drought seasons to rainy seasons, that you would see different populations would be better suited to be able to survive in that type of environment. And so in the dry seasons, birds with bigger beaks would survive because they only had larger, harder seeds, and the bigger beaks were capable of breaking those down. And then within the wet seasons, you'd see birds with the smaller beaks. Okay, birds with the smaller beaks were able to eat the smaller seeds. And so the proportion of small beaks would increase in the wet seasons. And so just within those times and seasons, you would see these populations thriving one over another. Now, there are two main theories that describe the rate of evolutionary change, specifically looking at macroevolution. So when we have the origin and creation of a brand new species, there are two theories for thinking about this. Okay, and those are gradualism and punctuated equilibrium. Now, gradualism explains evolutionary changes that lead to the creation of a new species as very slow changes happening over time that ultimately lead to the rise of that new species. So ultimately, within a generation or even generation to generation, those changes really wouldn't be perceivable. So the changes are looking at over the course of a longer time frame of millions of years. Okay, so now looking at punctuated equilibrium, a very different theory. So here you are looking at long periods of time with very few evolutionary changes. So that would be known as a period of stasis. Okay, so you can see a period of stasis here. And then some sort of event would occur that would cause for a brand new species to evolve. Okay, and so you can see here periods of stasis followed by very abrupt evolutionary changes occurring within populations. And then periods of stasis followed by very abrupt periods of evolution. Okay, so very different contrasting views here. The theory of gradualism, where extremely slow changes occur over time, leading to the rise of a new species. And then the other, where you have periods of stasis followed by very abrupt bursts of evolutionary change. Now, in providing evidence for macroevolution, evolutionary biologists look to three main things. So they look at the fossil record, they look at homologous anatomy, as well as homologous molecules. Okay, so remember the term homologous. When we looked at homologous chromosomes are those that are similarly in terms of what genes were located on them, but not identical to one another. So when we're looking at homologous structures, we're talking about structures that have maybe a similar type of function or they're located in a similar region, but they don't look identical from species to species. Now, just to give you a few really cool animals here, we've got the alligators as well as the hagfish. Now, the hagfish species is over 500 million years old, and crocodiles about 230 million years old. And this is based on looking at the fossil record, and so the earliest evidence of that species existing. Okay, so some of the more natural selection pressures, you've got predation, okay, so predators, as well as infections within a population. Okay, if certain individuals are more suited or more resistant to a certain type of infection, they are going to be the ones to survive. Now, toxins, climate change, as well as habitat change, this is where we're getting into looking at human impacts on the environment, right? Because the introduction of certain toxins into environments can cause for either the food source to be depleted or for the negative impacts on the population that exists in that region. With climate changes, you're seeing changes now in topography. You're seeing topographical changes. And so we have issues there with certain species not being, you have changes occurring in the Antarctic, right? With the ice shields. Okay, with the reduction in ice, well, that is the habitat for the polar bears. And as that habitat reduces due to climate change, Okay, and so then as a result, you start to see that the population sizes start to decrease. And one of the things we as humans do then to try to preserve species nowadays is utilize captive breeding programs where you are providing an opportunity for that species to be able to reproduce and ultimately to continue on. Now, okay, there are a lot of ages listed on these next few slides. The ones that I want you to pay particular attention to are those that are bolded. Okay, so the Earth, roughly about 4.5 billion years old. We have the first appearance of prokaryotes at 3.5 billion years ago. Eukaryotes with our single cell protists, first evidence of them, it's about 1.5 billion years. Mammals coming into play at about 200 million years ago, and then Homo sapiens about 200,000 years ago. So if you consider 4.5 billion years to 200,000 years, we're looking at a very small time in terms of the history of the Earth that we've been around. Again, this is showing you a way in which they would take a look at different fossil records of different skull sizes and types and shapes and be able to map out the evolution of a given species. And so here we're looking at a range of primate skulls that have all been plotted here. And so they have arranged them in terms of brain capacity and then in terms of dating from current back to that 4 million years. And so we have a range of skulls here from modern chimpanzees, gorillas, ultimately human skulls. And viewing the various shapes of the different fossils, you can see how they've arranged to show a trend in changes to indicate a likely common ancestor between humans, gorillas, chimpanzees. Okay, and so evidence for macroevolutionary theory comes down to these three places: the fossil record, the anatomical record, looking at homologous structures, as well as the molecular record, looking at, as we've been able to increase genetic technology, taking a look at various similarities between current populations and ancestral genetic information. Now, with the fossil record, it's one of the most direct evidence of evolution. And the great thing is that bones contain calcium. Remember, it's a huge reservoir for calcium. So regardless of species, their skeleton's made out of bone, and this highly mineralized extracellular matrix cannot be degraded by microorganisms. And so what happens is the bones ultimately mineralize to form rock. So other fossils, you can see feathers, scales, hooves, horns, teeth, shells, other impressions. Here you can see some remnants of feathers that exist within this fossil here. Now, I'm sure we've all seen Jurassic Park, where they proposed that they could extract dino DNA from the blood that the mosquito had before being encased within the sap of a tree. Now, the problem with the fossil record is that it is a very rare occurrence and conditions must be perfect for a specimen to be preserved. And so as a result, the fossil record is relatively incomplete. Okay, there's lots of huge gaps. So if we come back here looking at these hominid skulls, you can see there's not a direct correlation from one to the next where you can see that gradual change occurring. You're having to fill in a lot of blanks, and that's just because the fossil record is relatively incomplete. Now, another place for evidence of macroevolution is through the anatomical record. And this is where we can look at homologous structures that exist between different species. So remember, with homologous structures, we're looking at structures that are similar between species here, but not exactly the same. And so a few examples here: invertebrate embryos during early embryological development, all embryos, looking fish, reptiles, birds, and humans, all contain these gill slits known as pharyngeal pouches. Now, ultimately, when that specific embryo develops further, they develop into very different structures. So in fish, for example, the pharyngeal pouches develop into gills. Whereas in humans, the pharyngeal pouches develop into a number of different structures that we can find within our head and neck. Also, within every vertebrate embryo, there is a bony tail. Okay, so in fish, reptiles, even birds, those tails remain, but in humans, those are vestigial structures that ultimately terminate at the coccyx. Lastly, human embryos develop fur during the later stages of development. So sometimes little newborn babies, they can have this little soft fuzz on their bodies known as lanugo. Now looking at homologous structures now between different species, here we have the upper limb of a human compared to the foreleg of a horse, a cat, and then the wing of a bat and the fin of a whale. All right, so all structures that essentially are like the arms of the human, right? And we can see there are a lot of similarities in terms of the bones that make them up. And so with the humans, we have the metacarpals as well as phalanges, which would be homologous structures. Okay, and you can see them identified here, the homologous structures between the different species, horse, cat, bat, and whale. And we can look through them, the carpals, the homologous structures here within the horse, similarly radius and ulna, and how you can see those represented in other species, and then also the humerus. So very different anatomical structures, right? Different landmarks, different shapes, but ultimately homologous in nature in that they form that specific region of the body. Now lastly, with advances in DNA sequencing technology, we're able to take a look at the molecular record, take a look at DNA sequences between different species. So here we're comparing human DNA to mouse DNA. And you can see for this one sequence of DNA, although we're completely different organisms, we have very few differences here that you can see identified between the human and the mouse. Really, we have a substitution there, all of the white nucleotides would be the differences. And so, remember, as I mentioned before, each triplet of nucleotides is going to code for an amino acid. Well, you can see in this case, we have the same amino acid. So the only proteins that are different are those that are identified in red. Okay, so very few differences, but enough differences to make proteins between the two species, humans and mice, very different. In week 1 of genetic inheritance, where we focused on monohybrid crosses, you could see there that your genotype frequencies for crossing two heterozygous individuals would be 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive. Now we're going to take a look at frequencies of alleles as they exist within a population. And there's a few different formulas we can use. Now we're going to be able to calculate the frequency of those given alleles in future generations. Now, two individuals, Hardy and Weinberg, they studied the frequencies of alleles in a gene pool. Now, these guys came around 50 years after Darwin had originally coined the origin of species and the survival of the fittest. They were looking at the different traits that exist within populations, but at varying frequencies. They found that if the proportions of a given genotype remained constant from generation to generation, that population was in a state of equilibrium. And we know that theory as Hardy-Weinberg equilibrium, named after the two individuals that described it. Now here's our first example. In a population, we have 1,000 mice. Now I'm wanting you to calculate what is the frequency of black mice and white mice if 600 have black fur and 400 have white fur. Okay, so in order for us to calculate the frequencies, this is similar to how you would calculate the mark that you got on an exam. Okay, so say your exam's out of 100 and you want to know what your percentage was. Let's take your grade. Let's say it was 75. You divide it by 100 and it gives you 0.75, right? You multiply that by 100 to give you the percentage, okay? But what you're actually calculating when you take 75 and divide it by 100, you're calculating the frequency of correct questions to total questions. So you can do the same thing here when trying to calculate the frequency of black mice and white mice in the population. So we have a total of 1,000 mice. We have 600 that are black, and we have 400 that are white. All right, so first off, with the frequency of black mice, we would take 600, divide that by 1,000, that would give us 0.6. And to calculate the frequency of white mice in that population, we would take 400, divide that by 1,000, which would give us 0.4. Now, it's important to understand that both of these frequencies must add up to 1. Now, knowing the frequency of the phenotypes, you can then calculate the frequency of the genotypes and alleles in a population, and that's what we're going to be doing. Now, when we're calculating these allele and genotype frequencies, we're going to designate the more common trait with the letter P. Okay, and the less common trait we are going to designate with the letter Q. And we're using these letters to be able to create a formula for ourselves. If there's only 2 alleles for a trait in the population, then we have to assume that the frequency of P plus the frequency of Q is going to equal 1. And so for our purposes, we're just going to be looking at traits where there are only 2 alleles. We're not going to make it any more complicated than that. Okay, so this is going back to when we looked at complete dominance, right? One was dominant and one was recessive. In this case here, the more common trait that we're seeing, we're going to identify as P, and the less common, Q. Now, the dominant allele that will be given will be designated by a capital letter, and the recessive will be designated by a lowercase. Okay, so say in that previous example, we had mice with black fur, that would be dominant, and those with white fur, that would be recessive. Now, a way you can look at this is that the more common is also going to be the dominant, and the less common is more likely to be the recessive. Now, of all the possible genotypes that you can create from your dominant and recessive alleles, think back to your monohybrid crosses. If we have complete dominance, one allele for black fur is dominant over the recessive for white. The 3 possible genotypes are homozygous dominant, heterozygous, and homozygous recessive. So we have 3 possible genotype outcomes. Therefore, in that population, we have 3 different possible frequencies occurring for each of these genotypes. So remember, previously we said we're going to identify the more common trait as P, and that can be associated with the dominant allele. And so our formula will be set up as such. P squared plus 2PQ plus Q squared equals 1. So let's look here. If your P is the most common, you have P multiplied by P. That would be your P squared. PQ, that's going to be the frequency of your heterozygous. Remember, we end up with 2 of those. And for the frequency of your homozygous recessive, Q multiplied by Q gives you Q squared. Okay, so if it helps you right now just to be able to translate from your genotypes to looking at the frequencies of those alleles, then you can write it out as P multiplied by P plus 2 times P times Q added on to Q times Q. Okay, and the frequencies of the homozygous dominant, heterozygous, and homozygous recessive will all add up to 1. And we're going to use these numbers to determine the frequencies of a given genotype existing based upon the frequency of the allele. So your 2 equations for this section are frequency of the dominant allele, most common allele, and the frequency of the recessive allele is going to add up to 1. And then we can calculate the frequencies of the genotypes, okay? So up here, label this as frequencies of alleles, and this equation is for frequencies of genotypes. So you can see here we have the P squared, so 2 of the dominant alleles, 2 multiplied by a dominant and a recessive, and then the recessive frequency squared for the homozygous recessive individual. All right, so let's take a look at our first example. I'm going to walk you through this step by step, and then we have some other examples in the lecture notes. Take a look at them, try to work through them yourself, but I'm going to go through each of those piece by piece during our tutorial together. Okay, so this example here that we're going to walk through, in a population of lizards, they have red eyes and green eyes. Now, the frequency of the red eye allele is 0.7. Okay, there is some key terminology we need to pay attention to. Remember, P represents the frequency of the dominant allele, and Q represents the frequency of the recessive allele. So in this case here, if I'm telling you that the frequency of the red eye allele is 0.7, then the frequency of the green eye allele must be 0.3, right? Because P plus Q has to equal 1. And so you now know the red eye allele is dominant and the green eye allele is recessive. Now, if my question is asking you what proportion of the population would you expect to have green eyes is the phenotype. And if you know that green eye allele is recessive, you need to have a homozygous recessive individual in order to have green eyes. And this is how we'll work through this question. So first off, we designate the dominant allele by P, and we know that the allele frequency formula is P plus Q equals 1. If we know that P equals 0.7, because that was given in the question, you know that Q, the recessive allele frequency, must equal 0.3. Now, this is the formula we are going to use to be able to calculate the phenotype that we were asked. So we were asked, what proportion of the population is going to have green eyes? Well, green eyes is only going to happen as a phenotype if the individual is homozygous recessive. So in that case, you're looking at your homozygous recessive frequency will be Q squared. So what is Q? Q is 0.3. So let's work through this. If you know the frequency of the green eye phenotype is Q squared, you have 0.3 being the frequency of the recessive allele. Therefore, 0.3 squared, or 0.3 times 0.3, gives you 0.09. So therefore, you would expect 9% of all of the lizards in a population to have green eyes. With the text and through your notes, try to work through the rest of the questions by yourself, and then we will go through them step by step live during tutorial. Now, as I mentioned before, with Hardy-Weinberg equilibrium, this can only occur if ultimately no natural selection is occurring. Okay, so no selection pressure on any specific trait. Also, a number of other criteria are required to be met. Population needs to be very large, mating must be random, there must be no mutations occurring, and no input of any new alleles. What does that look like? There can be no new individuals added to a population from outside of that population. So ultimately, the Hardy-Weinberg equilibrium is referring to a population where all of the above assumptions are true. Now, the population in this case then has to be stable. There can be no evolution occurring. Okay, and then those allele frequencies also, you're assuming they're going to stay the same. So the example of population genetics that we looked at, as well as those that we'll cover during tutorial, those are assuming the population is in Hardy-Weinberg equilibrium. Okay, those calculations only work if you do not have any of these following. And so the way that those population genetics calculations and assumptions can only work out is if a population is in Hardy-Weinberg equilibrium and meeting all of these criteria. So when a population is not in Hardy-Weinberg equilibrium, we're going to take a look now at what those agents of evolution are. Okay, so we're going to look at the following 5 factors that are capable of altering the proportions of homozygotes and heterozygotes enough to provide significant deviations from otherwise what the Hardy-Weinberg equations would predict. Okay, and those are looking at mutations, gene flow from one population to another, non-random mating, genetic drift, and then ultimately selection pressures. So first off, we're going to take a look at various selection pressures that can occur. Right? These are 3 examples of natural selection. We have directional selection, stabilizing selection, and disruptive selection. So first off, the upper row is showing us a distribution within a population for tail length. Okay, and so we're looking at our normal bell curve here. This is showing you the distribution before that specific selection pressure had occurred. So in this example here, we're going to take a look at directional selection. Now with directional selection, there's one extreme trait that is going to be favored over another. Okay, so in this case here, we're looking at the longer tail length being more favored than the shorter tail length. And so what you're seeing here in these blue regions are the lengths of tails that are not selected for, okay? So the longer tail length in this case is being selected for, okay? It's more advantageous. And so what happens with directional selection is you have a shift in the overall population where the average tail length will move towards that extreme that's being selected for. And so in this case, we are selecting here for longer tails. They're more advantageous for whatever reason. When natural selection is acting on that population, you're going to see that those with longer tails are going to be the ones that are more likely to survive. And so you see a shift where the average number of individuals in that population will now have longer tails. Okay, if we were to select for shorter tails, you would just see this shift happen in the other direction. Okay, so directional selection, you have one extreme being selected over the other extreme trait. In this case, we selected for long tails over short. Now with stabilizing selection, what you see here is an accentuation of the mean, right, of the average in a population. And so with stabilizing selection, what we see is that it is the mean with stabilizing selection, what we see is that it's the average trait that is actually selected for. And so in this case here, it was average tail length is most advantageous. And so as a result, you're going to see more individuals with the average tail length that will be selected for. And so you'll see an increase in that average. Okay, so that's stabilizing selection. Now with disruptive selection, we actually have a situation in which both extremes are selected for. And so what happens as a result there is you see increases in both extremes, but you see a definite decrease in the average tail length in this case. So let's take a look at some other examples here. Remember, with directional selection, it's one extreme over another. So we're selecting for one extreme trait and we're selecting against the other. So in this case, let's just say the long wiggly tails, they looked like a snake and they would scare predators. And so that was the trait that was selected for. The longer the tail, the more it looks like a snake. And so that trait would be selected for. That is directional selection. And so you have the overall average of the population shifts in that direction. Now with stabilizing selection, in this case here, we're looking at short and long tails. But here with short tails, they mess up the cat's balance. So that's not good. And with long tails, they drag on the ground. So that's not good as well. So it ended up in this case, we have a medium tail for the cat that is perfect for their environment. And so ultimately the selection is against both extremes and for that intermediate average. Okay, and so ultimately the moderate trait in that case of medium tail length, it will be selected for, and you have stabilizing selection occurring. Now here we have both extremes being selected for. So short tails were actually great because they helped to keep predators from catching you when you were on the ground. But then when you were up in the tree, the long tails were also beneficial because they would help balance. Okay, whereas the medium tails just don't help at all. So in this case of disruptive selection, we have selection, both extremes are good, whereas the intermediate trait is not beneficial. So you have disruptive selection. So you can actually look at that stabilizing and disruptive are kind of opposites of one another. With stabilizing selection, it's the intermediate that's selected for. And with disruptive selection, it's the extremes that are selected for. Now, mutations also play a role, right? We know that our environment dictates a lot of what happens to our DNA. Now, in a short period of time, mutation rates are generally too low to significantly alter Hardy-Weinberg proportions, but ultimately the mutations that occur within the gametes, well, they can be passed on if they're not corrected to the future offspring and ultimately throughout the generation. And so ultimately, the source of variation within that population is going to occur over the long term. And so mutations can play a role in altering those allele frequencies over generations. Now, typically, random mating is going to occur in population where there's no preference for a certain gene for reproduction. Like with plants, for example, a flower is going to pollinate nearby flower just because they're in close proximity to one another. There's nothing about the genes of one that are going to dictate whether or not it's going to pollinate another. Now, in the animal kingdom, random mating rarely occurs, and that's because animals have preferences. And so genotypes are typically linked to reproductive behaviors. You have dancing rituals, strength. We saw those images earlier of those two rams that were headbutting one another. And also within our own human populations, mating is a completely non-random process. Right? There's so many different observable traits, cultural, play a role as to who is selected to be your mate. If a population is completely isolated, there will be no new alleles entering into that population, aside from those created from chance mutations. Now with migration, you have then individuals from population B can move over to population A, And similarly, those from population A move over to population B. In this example here, though, we're looking at in population A, we had selection pressure against the recessive phenotype. So as a result, you ended up with a population of individuals that were homozygous dominant. So now you have those homozygous dominant migrating to that recessive population of B. Now their phenotype is going to start to take over in that new population. This is just to show you that migration of individuals from one population to another can have a variety of impacts. Now, the magnitude of the effect of migration on that other population receiving the individual is determined by two main factors. You have the number of individuals actually migrating over, and then you also have the differences in allele frequencies between the migrants and the original population. So if you have a whole lot of migrants moving over, then you're going to see that difference in allele frequencies occurring and ultimately more likely to have the phenotype of the dominant expressed. Now, genetic drift is the process of random changes in those allele frequencies, not due to the overall fitness of that organism, but instead due to some sort of bottlenecking that takes place. Now, the bottleneck itself is some sort of event, typically a major environmental event, that would drastically reduce the original size of the population. So here we're looking at two different colored marbles, which are representing the alleles that exist for a certain trait within a population. So now, if we have that bottlenecking effect, which is going to drastically reduce the size, let's just say, a major wildfire or a major hurricane, something happened that just wiped out a population. The allele frequencies that existed before are not necessarily going to be the allele frequencies that exist when this small sample of the population survives. And it could be too that certain types of individuals for a certain trait were more likely to survive. And in which case your new population created by those surviving individuals is going to display a much different allele frequency in those future generations. So ultimately, the bottleneck effect is looking at a specific environmental change that occurs that reduces the population size significantly, and the allele frequencies that exist within those surviving individuals that are then passed on to future generations will look much different than its original parent population. Now, we talked about bottleneck events being mainly environmental, but human pressures from hunting can actually in and of themselves be a bottleneck. With cheetahs, they've had at least two genetic bottlenecks that have occurred within the past 10,000 years. So as a result, the genetic variability that exists within the cheetah population is very low. And remember, anytime genetic variability is low, it means that your population as a whole, very similar. And if any sort of environmental change were to occur, you will not have enough individuals that are going to be suited to that new environmental condition. And so risk of extinction is very high in those populations, looking at specific species where genetic variability is low. This example here of the Trinidadian guppies is looking at populations of guppies, which are little fish you can see here. Now, within these two different pools, we have two different populations of guppies, which look very different. And this is a prime example of microevolution, where you're seeing differences within these populations due to a variety of selection pressures. Now, in the pools above the waterfalls, the guppies are found along with a specific type of fish known as killifish. And these will sometimes eat the guppies. But in the pools below, there are a lot of pike, and the pike regularly prey on the guppies as their primary food source. And so you can see here the predation pressure in that lower pool below the waterfall is going to be much higher. As a result, guppies in those high predation pools are not as colorful as guppies in the low predation pools. Those with the genes that code for more camouflage type of scale color are going to be more likely to survive and pass on those genes to their future generation. And so less colorful means you're going to be more fit for that environment. Then also guppies in those lower high predation pools, they also tend to reproduce at a very early age and attain relatively smaller adult body sizes. If you're smaller, you're harder to catch, and reproducing at an earlier age means you're going to be more likely to be able to pass on your alleles to that future generation. Here you can see that the high predation pools are providing a specific selection pressure that is causing for specific traits to be expressed over others. And that brings us to the end of our lecture for this week. So remember, in tutorial, we will spend some time looking at population genetics and those specific examples in much more detail. Okay, so give them a try on your own and come up with your questions so that we can work through those together as a group. Now, if you have any other questions that come up in the meantime, please feel free to reach out. Otherwise, I look forward to seeing you all there in tutorial. Have a great week. Take care.